right. Welcome, everyone. Thank you so much for joining us today. My name is Sam Blickan, and I am the co-director of the Zooniverse crowdsourcing platform. And I'm going to be moderating today's event in my capacity as a board member of WeDigBio. So for those who aren't familiar with this event, WeDigBio is short for Worldwide Engagement for Digitizing Biocollections. And it is a global data campaign, a virtual science festival, and a local outreach opportunity all rolled into one. And this is a biannual event. So we host it twice a year, typically running across 4 days, once in the spring and once in the fall. And these events mobilize participants to create digital biodiversity data through virtual and on-site events. So as I said just a moment ago, this event is being recorded and the video will be made available on the WeDigBio website. So today's symposium, How is AI being used in biodiversity efforts? Will cover a range of perspectives on this admittedly quite broad topic. We'll have 3 speakers, followed by a Q&A session at the end of the hour. And if you have questions, you can use the Q&A function. So there's a Q&A button along the menu at the bottom of your screen. So you can use that function to share your questions, or you can post them in the chat. I will now introduce our first speaker, Dr. Sarah Huebner. Sarah is an ecologist and program manager with the Smithsonian's National Zoo and Conservation Biology Institute, where she coordinates the efforts of a coalition of organizations that are working together to deliver conservation technology solutions at regional, national, and global levels. Her research focuses on the practical aspects of conserving wild mammal populations, including long-term continuous monitoring to safeguard biodiversity. She's also an active proponent of employing citizen science and machine learning for conservation and ecological research and democratizing access to artificial intelligence. Her talk today is titled The Evolution of AI as a Tool for Conservation. Sarah?
Thanks, Sam, and thanks for having me. So every day, wild living animals must make crucial decisions for survival and for their reproduction in heterogeneous environments. Human-induced biodiversity loss, habitat loss, and climate change have dramatically increased this variability, introducing new challenges to accessing resources that may make prior behavioral strategies less effective. And with their behavior, wildlife in turn influence their ecosystems through foraging, migration, and reproduction. In some cases, animals like elephants quite literally reshape their environments by toppling trees and creating new paths through brush, opening up new niches for other mammal species. However, as the human population and associated global impacts continue to expand, elephants are forced onto ever smaller pieces of land and cut off from their traditional migration routes, some of which have existed for centuries. Many wildlife species exhibit novel, often maladaptive behaviors when they're forced to find new resources or migrate further from their ancestral homelands. So, in order to understand the status of populations around the world, ecologists urgently need to see when and where animals occur and evaluate their success in new environments. To achieve those ends, over the past decade, a diverse array of sensors have been deployed to monitor wildlife and habitats at unprecedented scales. These sensors are an improvement from traditional data collection methods, which involve walking transects or flying helicopters to count animals. Both of those methods were time-consuming, expensive, and typically resulted in undercounts due to animals avoiding humans on foot or in the air. But using passive sensors, we can collect data on wildlife 24/7, 365 at a fraction of the cost and in a minimally invasive manner. The most widely used sensors include wildlife cameras, what we would call camera traps, that record images and videos, GPS and radio collars that provide hourly location data on wild animals, bioacoustic sensors that record vocalizations and can be used to triangulate positions, and drones to immediately locate herds, prides, and poachers. So these technologies are great, but research teams around the globe set up studies with very different methodologies, making it difficult to combine data for comprehensive analyses. So one of the things we really need in this moment are ecological monitoring networks at regional, continental, and global scales using the same protocols to standardize data collection and analyses. To meet this challenge, I founded and direct a collaborative network called Snapshot Safari. So it's an ecological monitoring project, which is a collective of ecologists, wildlife managers, citizen scientists, data scientists, local community members, and other academics, all working together to monitor and protect African mammals. Since 2017, Snapshot teams have deployed more than 50 camera trap grids in protected areas in 7 Eastern and Southern African nations. Our motion-activated cameras run continuously, allowing us to evaluate African wildlife on an unprecedented scale, both spatially and temporally. We capture around 80 mammal species in most of the nature reserves where we work and many large birds. Every site uses standardized protocols to facilitate cross-site comparisons, which allows us to answer questions related to animals' distribution and density, including where they spend most of their time and how the presence of humans or other animals affects behaviors like foraging and vigilance. So you may be asking yourself, how much data are we talking about here? With nearly 2,000 cameras in the field maintained every 8 to 10 weeks, we have collected and classified more than 22 million images of animals in the wild over the past 6 years. We presently average 3 to 4 million images per year, and this is just one network. We need to scale up to networks like these all over the world and combine different sensing modalities to thoroughly understand what's happening with biodiversity. The amount of data we're producing means that we need to turn to citizen science and AI to label and compile all of the data points. Our flagship project, Snapshot Serengeti, began in 2010, and it was the first ecology project on Zooniverse, making a very early entry into the field. Because of this and our policy on running cameras year-round, which has resulted in a massive image library, all of which have been labeled by Zooniverse volunteers, our data has provided the basis for more than a dozen AI models, including incorporation into Microsoft's Megadetector and quite a few species classifiers. Because no relevant models existed when we first decided to explore AI, we worked with machine learning developers to create our own custom object detector and species classifier, both of which are convolutional neural networks, or CNNs, which are the models most commonly used in ecology and conservation. CNNs are basically a set of layers stacked on top of each other. The first layer might look for very simple objects like the shape of an animal. In the next layers, the AI starts to understand more complex features like the spots of a leopard or the shape of an elephant's trunk. And in the final layers, the AI starts making decisions based on what it has learned. For example, providing a determination of the species based on what it knows. One of the challenging aspects of training CNNs is the number of training samples needed to teach AI to accurately identify the targeted feature class, such a species, typically is on the order of several million images to train a model. So one of the realities of motion-activated camera traps is that despite efforts to cut back vegetation, at some point you will have grass and tree branches setting off the cameras, resulting in images with no animals in them, which are labeled as empty for our purposes. In many camera trap data sets, these empty images can make up as much as 80% of the captures, which means a lot of slogging through empty pictures to find the animals. So this is the first step at which the use of AI can really help researchers who need to evaluate their data quickly to inform conservation management programs. Object detection CNNs are able to identify empty images as such while placing bounding boxes around the animals found in evaluations. Megadetector is the standard object detector most ecologists use for handling this task, and you can see the output on the left, in which it has localized animals with a high confidence score without attempting to name the species. The Snapshot Safari object detector goes a step further in identifying the species it has spotted, even when multiple species have been captured in one image, as you can see on the right-hand side. However, I should note that even with models trained solely on our data and stationary camera trap stations that stabilize the background habitats, there is a decline in accuracy and recall in later batches of data, as you can see in the table here. This means that we must continuously update and educate our models by adding more labeled data or transfer learning to other snapshot sites. Once we've identified and dismissed the empty images, we're left with the task of annotating various data from the wildlife images. Since its inception, Snapshot Safari volunteers on the Zooniverse have recorded as much data as possible from our camera trap observations, including species, counts, age, sex, and observed behaviors of all visible animals. As a result, we were able to train a species classifying CNN that returned predictions on all of the classes within each of those parameters. It performs very well on identifying species, fairly well on counts, and somewhat decently on behaviors and demographics, since those are harder to classify, even for humans, so the training data is not always easy to interpret for AI. To date, Snapshot Safari is one of only a few conservation projects actively using AI within our data processing pipelines, and that's because there are a number of issues that make it difficult. This figure is taken from a paper we published on combining citizen science and AI. These are all examples of images that are difficult for algorithms to accurately classify. Starting with the honey badger there in A, we don't have very many images of them. They're rare within the data set, meaning we don't have a lot of training samples. B and C show elephants in different backgrounds, which is very common when you're looking across 50 different sites. D shows multiple species, which is something that AI struggles with. Also, large counts. When you have a whole bunch of animals herding together, it can be difficult to sort out how many there are and what behaviors they're exhibiting. In F and G, you can see animals that have feeding that looks very different depending on the species. And in H and I, you see demographics of age and sex, which none of our models are currently able to do. So what that means is that we still need human supervision of all our classifications, or what is called humans in the loop. In a forthcoming paper, I calculated the gains in accuracy of incorporating humans in the loop and found that the overall error rate of about 35% by AI was reduced to 8.73% across the board. Humans outperform machines in 95% of the species classes. Our species classifier performed best on common species, for which we could show it thousands of images in each species class, but there aren't all that many of those. So, what are the future applications of AI and technology? There are a couple of next generation projects in the works using Snapshot Safari data. One that is really exciting is identifying individual animals within populations. As you can see by this guy, elephants love to scratch their ears on trees. This leads to tears and holes in their ears that last a lifetime that we can use to identify them repeatedly on camera traps to see where and when they are spending their time, which tells us how they are adapting to human-induced activities. So you can probably see that these are not the same elephant, but it's tough for humans to re-identify individuals simply from images. And this is where some of the newer AI models excel. They can learn the features of each elephant's ears and recognize them again every time that they see them. We recently ran a project on Zooniverse where we asked volunteers to trace elephants' ears and report on various types of features on each elephant, including whether their tusks were missing or broken, and any significant scars or markings on their bodies that make them distinctive. The volunteers' responses provide data that we will use to train a CNN to identify elephant individuals in a population in Botswana. Besides elephants, we also have the potential to create individual recognition models for cheetahs, giraffes, leopards, and other animals that exhibit unique markings. So even lions aren't out of the question if we can get enough good images of their whisker spots, which are their unique identifier. So as I mentioned earlier, our species classifying CNN attempts to identify behaviors observable in images, but with middling success. Behavior is a really important component of conservation programs, and AI models have developed to the point that we can now apply different methods to extract that data. So if you've used ChatGPT or the AI on your phone, you're working with a large language model or LLM, which scoops up data on the internet and returns summaries. LLMs aren't capable of accurately identifying what's happening in camera trap images, though. So we recently submitted a proposal to NSF asking for funding to create a series of vision language models in which volunteers would help us with different types of tasks. VLMs look at images and captions related to them to learn context and understand what is happening in a photo. So one workflow to help with this would be to view a grid of images and confirm or reject whether they fit within a particular query. So here I am looking for images of hyenas eating. Most of those were correctly identified that way by volunteers on the first pass, but one of these is of a leopard with its dinner, and another, the hyena, is not eating. So this would be a refinement task to ensure that the data we give models is accurate. And another fun task that we'll ask volunteers to do is to create their own free-form captions for images with as much detail as possible. So we'll then ingest all of the different permutations of volunteers' responses into the vision language model to teach it to understand context and annotate multiple categories from a single image. In the picture on the left, though most of these animals are eating and exhibiting minimal vigilance, indicating a sense of security, there are also animals moving and standing. So segmenting each animal using bounding boxes is inefficient due to overlapping edges. A description of the herd's overall behavior is more useful here to teach the machine context. On the right, this image would presently only be labeled with the three species and the corresponding behavior categories. We collect the information, but it wouldn't flag for us that this is a unique picture. So though the tortoise and bird would be labeled as resting and the baboon as moving, a traditional AI model could not interpret this event in a meaningful manner. So we're very excited about some of the things coming down the path. And I'll leave you with a final comment on the role of community science going forward. AI models are being trained to do the mundane tasks that volunteers did in the past, freeing people up for new, more challenging tasks that will help ecologists determine how wildlife are faring in the face of climate change. So we will absolutely keep working with all of the great people on Zooniverse to continue to advance the role of technology in conservation and biodiversity monitoring. So these are some of the partners we're working with at the Smithsonian Institution to advance applications of conservation tech and enable us to efficiently track wildlife populations now and in the future on a global scale. If you're interested in helping out with our first pass data labeling, I plan to introduce more data to wildcat Gorongosa on October 25th. Thank you.
Thanks so much, Sarah. That was wonderful. Just as a reminder, folks, we have a Q&A feature on the Zoom. So even during the talks, please feel free to drop questions in there as they come up. And then at the end, during the Q&A session, we can pull from those questions. So moving on, our next speaker is Dr. Daijing Li, an assistant professor at University of Arizona's Department of Ecology and Evolutionary Biology. He's interested in understanding how environmental changes have and will affect biodiversity through combining empirical and quantitative approaches. His talk is called Phenobase, a central global database of plant phenology by integrating in situ observatory and image-derived phenology data.
All right. Thanks, Sam, for the nice introduction. And let me share my screen. Can you see it?
Yes.
Okay, great. So you're seeing the full scoring one, right? Okay, awesome.
Yes.
Yeah, great. I think Sarah did a excellent job and kind of introduce how machine learning or AI can be used to monitor wildlife. And here I'm kind of actually switch to plants. And I would argue, it's also very exciting to lots of exciting things we can do for plants. And here I'm just briefly talk about a project we're working on to compile a global database of plant phenology by integrating the in situ observatory data and also data from images. So why technology? Here we can see beautiful pictures about flowers and fruits, or even just some of them, mostly with leaves. So that's kind of, plant phenology, the timing of this key life history events, right? Like leaf out or flowering or fruit. And they have important consequences for both ecological and economical reasons. For example, the timing of the leaf out will affect the growing season and will affect carbon cycle as well as the interaction with local climate. While the timing of flower or fruit not only have important consequences for the fitness of the plants themselves, but also affecting the species like depends on the plants or interact with the plants, right? Like pollinators. And the time is critical because let's say if the important like crop, like the peach trees, they flower early because we have warmer and warmer spring. And later, if we have a frost, then most of the flower probably will be damaged. Then we're gonna have lots of economic loss. So they all have like very important consequences, and I think that's why I think study plant phenology is critical and so where we are in terms of connecting plant phenology data. So largely we're talking about two types of phenological data. So the first one is from human observations, like the in situ observations. In the US, we have national phenological networks. And also like all the countries have a different type of networks too. And we also have specimens, right? Like with the digitization of specimens, we also have lots of potential data there. And but on the other hand, we have remote sensing data from satellite imagery and also like phylocam, for example. So that's kind of largely the two types of phenological data we have. And here, I would just mostly focus on the human observation data here. So over the past 2 years, like, after working on multiple projects of plant phenology, we realized the current plant phenology data has lots of, limitations. For example, the first one would be the incompatible of the existing phenological data. Like as I mentioned, in the US, we have this National Phenology Network, and in Europe, we have this Pan-European Phenology Network, and all the countries like, Africa, South Africa or Indian Brazil, they also have the national or regional phenological networks to monitor plant phenology. But this different, organizations, they use a very different way to describe phenology. So even though we have, let's say, if we have all this data, it's just not easy or ready to just use them, right? You need to somehow make them talk with each other to kind of integrate them. So that's kind of the key limitation on that side and the existing. And then the second limitation is like we have this large spatial and taxonomic gaps. So, for example, here's a map, with the existing open or semi-open sort of plant phenology data, like mostly in the Europe and the United States, but other countries. They probably have some data available, but they're not open. And again, the we cannot just grab them and use, right? And also we can see like just like as other things, very very common patterns like lots of records belong to a small number of species, right? So lots of species we just simply don't have any phenological data. And third, we also have this huge historical gap here. Like if we look at the pattern of the existing data we have here, it's just like most of the data actually were from after 2010. And before then, we just like have very limited number of records across all species, actually. So that's kind of the key limitations we realized. But there's also potential, solutions for this limitation. So, for example, the first one, I think we can developing like, the like ontology, which is a kind of, a scaled or technology to organize knowledge, right? So if we can do this and integrate all the existing data, that it can easily provide us like more than 30 million of records of existing plant phenology data. And the for the second limitation, I think iNaturalist or other kind of a citizen science or community science program, we have the data there, right? If I upload a plant image, very likely it has information about leaf or flower or fruit, right? So we have the information there, and if we can somehow kind of extract this information, then that can give us lots of the data. So here in the map, um, so this is like showing all the plant observation from iNaturalist. I would say, it's actually pretty well covered spatially, right? Compare with like the limitation here. So if we can extract those information from those images, that would be great. And so far, like, we have more than 50 millions and increasing rapidly plant images from iNaturalist. And also like, about 5 million of them have been annotated by iNaturalist users. If you didn't know this and think, iNaturalist, you can actually go to the website, or if you use the Android version of the app, you can actually annotate plant phenology from images. So if you haven't tried this and if you're interested in, I highly recommend to do this, and it's going to be fun and also contribute lots of training data for machine learning models later. Okay. And similarly, if we can use machine learning to analyzing the digitized specimens, and then we can have like easily 50, 60 million records, and this will cover hopefully the temporal gap here. So then the question would be, can we actually do this, right? Can we scale up using the machine learning technology to extract those information? And if a success, I think it's going to be a huge saving of our work and providing important data for us. And so our initial kind of a pilot analysis suggests, yeah, we can actually do pretty good job using the machine learning to look at some kind of wire annotated genus. But the big question is like, can we actually scale it up to all angiosperms? Like, let's say, what about 15 million images from iNaturalist? Everything like, when we scale it up, it's just like very different from what we deal with. We have a much smaller number. Um, so I think the short answer is yes, it is very promising. I think we're developing this we call vision to actually automate and annotate images from iNaturalist, and we actually just going to we just submitted to the bioRxiv the paper, so it should be online pretty soon. So here I will just quickly just walk through like some of the performance or the data we get. And here is just like, the expert annotation. So we look at the presence, absence, and for here the column numbers were from the final version models. Like, we can see we have like 97.5% of positive, like true positive. From our validation data, and also 92.3 of true positive fruit data. And the true negative ones also very, very good. So 97.5 for flower and 96.7 for fruit. So the model is performing very well in terms of, So there's like a true positive and true negative. And here we actually purposefully try to make the true positive. That's our priority, right? We want all the kind of detection data have high accuracy because that's probably even at the cost of tossing lots of images that we are not sure, right? So maybe for those type of images, we actually, have a huge fraction of them actually have flower or fruit, but just like given the model we have, we just weren't so sure. So we'll focus on making it to have the true positive to our priority. Okay, so that's the first version of Vision model, and we generated over 30 million records. We saw 25 million of them to be flower and about 5 million to be fruit. And so this is like from about 119,000 of species from 10,000 genera and also about 408 family. And here it's just like the phylogeny of the genus, and the red bar are the existing human annotation data. And while, the blue ones were just the machine annotated data, they are relative proportions, so the percentage. So you can see for most of the genera, we actually, with the phenovision, the phenovision contribute to most of the phenological data now for those genera. Okay, so and here is like the increase in spatial coverage. So the panel A here, the bluish, light blue cells, they were um, areas we have at least some human annotated phenological data, right? So not surprisingly, mostly in the United States, Europe, and some coastal area. Um, and but like with the Phenovision, we actually increase the spatial coverage a lot. Like, tons of like, all these red cells were just previously there's no human annotated data existing from iNaturalist, and now Phenovision actually provide data from for those areas. And then the remaining ones were just the total number of machine annotated record and the proportion of those. And we can see most of them with more than 90%, or even some of them 95% of those phenological data now comes from machine learning. That's not surprising, but also kind of, very promising to actually move our phenological study to kind of across scales and probably looking at global patterns, things like that. Okay, of course, there's lots of challenges like Sarah mentioned for these models. And here, because of the time, I will just skip it and happy to talk about it later, in the Q&A. But we're also working on other parts like, how to integrate in the existing data, how to convince different data providers to actually share the data, right? So that's actually a very important thing. And how do you credit them back? Um, and we're also working on machine learning models on the digitized specimen. So stay tuned for that. Um, and putting all this together, so we have like this in situ data, and then we have machine learning data from images like iNaturalist or GBIF or WeDigBio, and putting all this together. So we're building this global database we call Finabase, and we have the website here if you wanted to know a little bit more about our kind of project. So you can visit it there. So with that, I want to thank you for coming, and we'll have a Q&A later.
Thank you so much. These have been really fascinating so far. Um, all right. So our third speaker today is Mike Trizna, who is a data scientist at the Smithsonian Institution in the OCIO Data Science Lab, though he is currently on a detail with the Chief Data Officer in the Office of Digital Transformation, which sounds very cool. Um, Previous to joining the Data Science Lab, Mike worked for 12 years at the Smithsonian National Museum of Natural History as a bioinformatician. I had to practice saying that job title. In his current role with the Data Science Lab, he collaborates with researchers and collection staff across the many units of the Smithsonian to pilot responsible applications of AI to their current work. So now I'll turn it over to you.
Hey, put this into full screen mode. See if this works. Okay. So thank you so much for that introduction. So I know that is kind of a complicated like job situating explanation there, but hopefully it will all make sense in the context of this. So today I'm going to be talking about leveraging ground truth Smithsonian data to evaluate AI tools. And compared to the two previous talks, I'm going to probably go back a step or go up a little higher level towards my current detail, which is focused on data and AI literacy and teaching that internally at the Smithsonian and also externally to wide audiences. All right, so the outline, I'm going to talk a little bit about my personal background from bioinformatics to data science, how they're kind of the same, and how they're different. And then this will be a little bit of an educational thing, going into the aspects of an AI project workflow, backed by some examples that I worked on with different partners from around the Smithsonian. Showing, I guess, first of all, like what an AI project workflow looks like, the importance of ground truth data, evaluations and metrics, data-centric AI, AI-powered data exploration, risk, and then share some resources at the end. If some of these terms don't mean much to you, hopefully we will address those. Okay, so starting off with my background, as Sam mentioned, I started out as a bioinformatician. I went to grad school to learn. I went to grad school in a bioinformatics program, and there I spent most of my time learning algorithms, how to implement them completely from scratch using programming. These are screenshots of books that were near and dear to my heart. I learned a programming language called Perl, which not many people use anymore. And I now use Python for the most part, but it was kind of a blast from the past finding these. And I do appreciate kind of like the kind of symbolism here that the O'Reilly covers had of going from a tadpole to a full-grown frog here. But then when I worked at the Natural History Museum at the Smithsonian, I largely was just using programming to stitch together data and using algorithms written and kind of optimized by others. I was hardly ever writing my own algorithms. That was kind of the progression there. And then when I switched over to working for the data science lab, at first, a lot of it was writing deep learning models completely from scratch. That was kind of the state of the art back then. It took a really long time to train these things. They were pretty brittle and they were really hard to deploy. But now I'd say that things have progressed where it's very kind of deja vu to where bioinformatics had moved, where largely using and adapting foundation models and kind of like doing lots of stitching together data input and output from one pipeline to the next. To go over kind of how an AI project typically works, this is actually taken from a lesson that I co-developed from with others at the British Library and the National Archives of the UK. And we put this into a library carpentry lesson, which I'll link to later. But anyways, what you typically will do is define what you're trying to do in your AI project. You will create your project team, gather data, figure out what you're trying to optimize using a metric. Then you will either use or adapt in existing model, then generate some results or predictions, and then you will assess the results. And then also, I don't even have on here like actually deploy this for people to use because honestly, we haven't gotten to that point yet. We're for the most part piloting things out, seeing if different approaches will work. But I guess that previous slide was kind of like, oh yeah, this is the ideal state, how you kind of like move from step one to step two. In reality, this is kind of how projects work in general. You start working down, you get like 2 or 3 steps in, you realize you need more data, and then you have to change your business need, or there's a million different circled backs. And this is more of like a spaghetti diagram, I guess. And then eventually you kind of like slowly work your way towards the end over time. Um, so yeah, just kind of like going through each one of those steps here with actual examples. I'll say that I've kind of alluded to this, but uh, there's this. I didn't really define what a foundation model is when I mentioned it before, but a foundation model is a machine learning model that is trained on a broad, gigantic set of data, such that it can be applied across a wide range of use cases. So think of ChatGPT. So like you can go to ChatGPT, you can ask it a question, and people are kind of like familiar with it being able to write things for you, but it will actually do most of like the natural language processing type tasks that data scientists have been doing forever. You can tell it to classify a bunch of reviews and see if they're positive or negative. These are things that people have been doing for a long time. Traditionally, it took you would collect a bunch of data, train your own model from scratch. But now these models are kind of able to do these things out of the box. Not quite as good, but they will do them pretty easily, and you can chain inputs and outputs together. So this diagram here talks about this new rise of this AI engineer position. So I think data scientists in the past had been training models completely from scratch, collecting data, and then refining that process. Whereas AI engineers are becoming more and more of a common task, who kind of will use ChatGPT to do something, and then refine the data either before or after, refine the outputs, and use it for useful tasks. Um, and this is kind of, if you are not necessarily in a data science position, um, and you're working on a specific domain, think of this AI engineer as something that you can kind of like apply to your existing work that you're already doing. So yeah, then also I'll say the next step here would be creating a project team. This is an example from a project that I worked on with the Smithsonian Transcription Center, and many others actually. So on applying AI models to transcribing audio recordings. And I wanted to highlight all of the people's names here, but I also really bolded the different affiliations within the Smithsonian to show that people were brought together, mostly led by Crystal Sanchez from the Smithsonian Digital Platforms team, to represent different aspects of the process, to represent different data sets, and different end users as well. So this is kind of like a great example. I've really enjoyed being on a part of this project, of bringing so many different viewpoints together to iteratively check in on progress of a project. And then gathering data is the next step. So I'll point out how important it is to have ground truth data to actually like validate if you're using something like ChatGPT or another model, how well it's actually doing compared to data that you already have, say, transcribed. So in this example here, this is actually from a very recent blog to the BHL, the Biodiversity Heritage Library blog, about how a large chunk of field notebooks from the Smithsonian Transcription Center was all of those transcriptions were sent over to the BHL database. And this is really useful because every time this topic comes up over and over again of like, can we use AI models to read handwriting in old documents? And the answer is sure, but like, how are you going to actually measure that? And you need to actually have the correct answer to start measuring those. So this crowdsourcing platforms are amazing for collecting that ground truth data to actually measure against. Yeah, and then how are you actually going to measure it? It's really going to change for depending on the task that you're doing. So again, this is going back to the audio transcription project that I was talking about before, where for that specific example, what you're going to want to measure using is the word error rate. So there is it kind of looks like a complicated formula here, but what you're really measuring is the number of mistakes that the model makes compared to the ground truth. And you can get one number for an entire audio transcription. But what's really interesting is that you can actually kind of like break this down by like the position overall in the overall transcription and look for peaks. So there you see a few large peaks here in terms of substitutions or deletions. And then you can take a look at the data and see like, why is it messing up at this point? What are the things that is causing the AI model to trip up? Or is there a problem even in the ground truth? That sometimes happens as well. So I will transition that then to this concept of data-centric AI, which is always relying back on the data rather than trying to make a better and better model. Seeing if your data needs some cleaning up, or going back to look for like misses to see if that reveals some sort of insight about how the model is functioning, or something that needs to be cleaned up in the data. So these are two examples of what are called confusion matrices, where I guess we took some digitized images of ferns in this one example. Here's another one with digitized fish, where we built a computer vision model to predict the species or the genus of the fish or fern. And if it got it right, you kind of get high accuracy and get purple marks here. But what's really interesting is that like when it gets it wrong, is it a close match? Is this something that's very closely related? And you do actually see this, especially in this fern example here. You can find these kind of like boxes that show close matches, and you would kind of bring these back to the botanist, and they would in this example they could tell you like, oh, I understand why I got this one wrong. We're actually not so sure if it belongs to this species or this species. So that's also part of an iterative process of like working, refining your model, throwing in additional examples to make sure you increase the accuracy overall. And then I'll also say that AI is really useful for exploring data in general. So you may have heard of the supervised learning versus unsupervised learning. Supervised learning is probably most of what you've seen so far in the presentations today, where you have a model, and it's trained on lots of labeled examples. And unsupervised learning, you may have seen the term embedding models, where you can kind of train a model to kind of like move towards correct answers or incorrect answers in directionality. These are two examples of models. One was used for kind of like grouping similar faces together. And this one is from a model that actually matches up chemical formulas with, I guess, images of bioimages of those proteins or something like that. I'm not really entirely sure, but I really liked how if it's the same, it moves it closer together. And if it's not the same, it moves it further apart. And what you can end up doing with these is throwing large data sets through, where you can actually start to see clusters. So like this is an example from a collaboration with the Biodiversity Heritage Library, where we're trying to find handwritten pages. And I recognize I'm kind of like running out of time here, but you get nice examples of like, here are all the handwritten text pages, here are the printed pages, here are the illustrations over here. Here's another one where we used it to match up herbarium sheets with specimens in in the collection using clustering, and another one getting into image descriptions to find different images that were described in a collection versus ones that weren't, and like, are there any that are close by that we could kind of skip a step? Um, that's kind of a review of the workflow. It's important to think about risk. I hate that I had to leave this to the end, and we'll kind of like skip over it, but there are good resources there. Um, and then additional resources, which I'll share these slides in the chat.
Thank you so much, Mike. That was really wonderful. It was a really great way to kind of tie everything together. It was really fascinating to see, even in three very disparate talks, where the sort of connective tissue lies. I know I saw one question in the Q&A, and we can start there, but I will encourage folks, if you do have questions for any of the speakers, or even just sort of general questions that any or all three could answer, please do add them. But we'll start with a question for Sarah. Um, and this question is from Fritz Pichardo. Apologies if I've mispronounced that. It says, "Hi, Sarah. The last part of the presentation with the image descriptions caught my attention. Is it possible to train the AI also with a sort of standard vocabulary or ontology that explains relationships that can be used then to link matches to the words used in the images by the annotations to that ontology? I wonder how that would work, if it's possible, or would it help the AI in understanding the images better based on the descriptions?"
Yeah, great question. I think that's one of the most appealing things about the new like vision language models is that it's really freeform and you can incorporate a lot of different types of data. And you're going to get, so if we have 20 different volunteers look at the same image, we're going to get 20 fairly unique combinations. And so um, that could look like a bear eating, or a bear eating at night, or a bear eating by a stream, or, so a bunch of different categories where you could get those relationships. And that is definitely something we're interested in, particularly in trying to understand predator-prey behavior.
Thank you. I don't see any more in the chat, but I do think that, we have about 6 minutes, but maybe this is an opportunity to dig a little bit into one of the topics that, Mike, I know you articulated. You were a little sad about having to gloss over some of the risk factor, which I'm sure all three of you could speak to. Um, I think I'm curious if each of you could just sort of give a very brief response about either uh, the risk mitigation that you've already thought about in terms of your project, or what you see as being sort of the predominant risks in the field. I know this is a big question and we only have 6 minutes, but I think I'm trying to get some lively discussion going. So anyone who would like to jump in here.
I'll, I'll start just because I kind of like brought it up by speeding through a slide. But I wanted to mention that using AI is completely different than how we've kind of like used digital tools in the past, because you have much less control over like what are the possible answers that it will give you, especially when you get into the generative AI type responses. And you're continuously given, I guess, the opportunity there is that you're given the ability to continuously tweak your model, to keep on looking for incorrect responses and then like go back and adjust. But it's really hard to kind of like predict what are the things that were like outside of your training set, the things that you didn't really think of, and how those might go wrong, and um, how your model will respond to those.
Yeah, I'd add to that. I think one of the risks always is making sure that your data is correct, because you can get a lot of different background noise, get a lot of different things that can be confusing. Um, so there's always that element of needing to verify that your data is accurate and your model's performing as you expect it to. Just another form of risk that is somewhat influenced by this, but looking at threatened species in the wild, you have to be very careful about where images go, and you don't want those to just be like out on the web for anyone to find with their location metadata. So a few different ways that risk can come in there.
Yeah, I would echo, totally with like both Sarah and Mike. I think, um, for our project, we thought a lot about like, how can we make sure that our data are kind of as accurate as possible, right? So that's why, I think in the talk I mentioned, we really kind of make that as our priority to make sure the positives are true positives, even at the cost of throwing out like lots of images, right? That we're not sure. I'm sure, out of those images, there's tons of positive, true positive images there. But just like, given our current model, we're not sure. So we would rather to have a smaller but good, accurate data than, a much larger data set, but with like lots of inaccurate data. I think yeah, I think that's a great point.
Thanks. In the 2 minutes we have left, I mean, we've talked a little bit about risk, and I mean, we could probably have another 3 hours just talking about that. I'm curious what the three of you are most excited about for these applications in the field. I think just a nice balance of, with great risk often comes great reward. So what do you see as the possibilities here?
Yeah, I'll lead off since I spoke first. There's a lot of really cool models coming down the pike, but I think the possibility of identifying individuals and populations and being able to track them over their lifetimes would really revolutionize a lot of the modeling that we're doing in ecology right now.
Yeah, totally. And also, I'm pretty excited about, what kind of new knowledge we can get out of, from the data we generated using the AI, by either kind of, extracting data from existing like images or text, or whatever, we can think about. It's just like the questions we would never had the opportunity previously to even kind of think about answering, right? It's just like fascinating to me. It's just to think about that, like what's the future there, it's kind of an imitation of the ideas.
And I'm most excited about hopefully figuring out this balance that I think Sarah and Daijing both mentioned about using AI as kind of like the first pass to get the really tricky problems to the experts, but being able to answer the really easy one kind of automatically and being able to just press the checkbox. So I think both mentioned it also to like to allow the experts to spend more time on the things that require expertise versus the things that are kind of more busy work to get at the scale that's required in some of these data collection things that are going on in.
Great. Well, thank you all once again. Everyone, please join me in thanking our speakers. It is 3 o'clock. Austin, I think, just put some information about WeDigBio in the chat if you're interested to learn more about everything going on. Today is the first day of our October WeDigBio event. It goes through the 13th, so please do head to WeDigBio.org and read more about some of the stuff that's going on over the next few days. But otherwise, I'll just thank you all once again. It was great to see you all and hear about the work that you're doing. So
Thank you,
have
Sam.
yeah, have a great rest of your day, everyone.
You too.
You.